Hi everyone. We just finished the 10 steps of glycolysis, but we haven't talked about how the cell keeps the final exit gate in check. Today, we're focusing on the regulation of our last irreversible enzyme, pyruvate kinase, or PK for short. We'll look at how your liver shuts this enzyme down during a fast to save glucose for your brain, and we'll explore the most common genetic defect in glycolysis, PK deficiency, and why it specifically targets your red blood cells. Let's dive in.
Here's a quick look at our roadmap for today's explainer. We'll start with the final exit gate, look at liver regulation during fasting, touch on long-term hormonal effects, jump into our clinical focus on PK deficiency, and then wrap it all up with some memory aids and a review.
All right, section 1, the final exit gate.
First, let's talk about how the cell coordinates the beginning of the pathway with the end. Pyruvate kinase is subject to a really cool mechanism called feed-forward regulation. Remember fructose 1,6-bisphosphate? That was the product of the master regulator PFK1 back in reaction 3. Well, when PFK1 is working hard, fructose 1,6-bisphosphate levels naturally rise and act as a direct signal to activate pyruvate kinase downstream. Imagine the whole pathway as a long assembly line. The first major machine, PFK1, produces a specific part. That part then literally calls ahead to the very last machine, pyruvate kinase, telling it to start running at max speed because a big batch of sugar is coming its way.
Conversely, PK is inhibited by ATP and alanine. Think about it. High ATP signals that the cell already has plenty of energy, while high alanine signals that the cell has plenty of raw building blocks. In both of those cases, we simply don't need to burn more glucose, so the gate closes. It's an incredibly efficient internal feedback system.
Moving right along to section 2, liver regulation during fasting.
Now, the liver has a very special requirement. During a fast, the liver actually needs to stop burning glucose so it can make glucose for the rest of the body, a critical process called gluconeogenesis. To do this, it uses covalent regulation. Here is exactly how that mechanism works. When your blood sugar is low, glucagon is released. Glucagon then binds to its receptor, which increases internal levels of CMP. That CMP activates protein kinase A, or PKA. And finally, PKA phosphorylates pyruvate kinase, which makes the enzyme completely inactive.
If we trace this hormonal cascade, glucagon triggers adenylyl cyclase to make that CMP. The CMP activates PKA, which literally sticks a phosphate onto pyruvate kinase. This phosphorylated version of the enzyme just cannot convert PEP to pyruvate, cleverly forcing that PEP to go backward into gluconeogenesis instead. And, as you might guess, when you finally eat and your insulin levels rise, a phosphatase removes that phosphate and pyruvate kinase is reactivated, ready to burn sugar again.
To really cement this for exam time, think of the glucagon padlock. Whenever you picture the liver during a fast, imagine glucagon walking right up to the pyruvate kinase gate and snapping a heavy padlock on it. It locks, or phosphorylates, the gate in the liver so glucose can't be burned, saving all that precious blood sugar for vital organs like your brain. Okay, section 3, long-term hormonal effects. All those allosteric and covalent changes we just talked about, those happen in minutes. It's rapid response regulation. But what if you consistently eat a high-carb diet? Well, your body performs long-term regulation. Instead of just flipping existing enzyme switches on or off, high insulin actually increases the gene transcription and synthesis of brand new glucokinase, PFK1, and pyruvate kinase molecules.
Over just hours or days, your liver can actually increase its glycolytic capacity by 10 to 20-fold simply by building more machinery. It's like a factory not just speeding up its current assembly line, but actually knocking down walls to add 20 new production lines just to handle that persistent, heavy carb load.
Let's pivot to section 4, clinical focus on PK deficiency.
Finally, we have to talk about PK deficiency. This is the most common genetic defect of glycolytic enzymes. And while the genetic mutation affects all your cells, the symptoms are almost exclusively seen in your red blood cells, or RBCs. Why RBCs? Because mature RBCs completely lack mitochondria. They are 100% dependent on glycolysis for their ATP. They desperately need that ATP to fuel ion pumps that maintain their flexible biconcave shape. Here is how the pathophysiology plays out. Low PK activity leads directly to decreased ATP production. Without ATP, the ion pumps fail, the cell loses its shape, and the membrane becomes damaged. These deformed RBCs are then identified as garbage by splenic macrophages and destroyed through phagocytosis. The end result is non-spherocytic hemolytic anemia. In severe cases, patients may even require regular blood transfusions.
A great memory aid here is the dying battery. Think of the red blood cell as a device that runs exclusively on the battery power of pyruvate kinase. Without the energy from PK, the RBC's battery dies. The lights, or the ion pumps, go out, the cell wilts like a grape turning into a raisin, and the spleen just throws it in the trash.
Interestingly, the liver is usually completely fine in these patients. Why? Well, liver cells actually have mitochondria for backup power. Plus, as we just learned, they can also just transcribe genes to make more enzyme to compensate. The poor red blood cell simply doesn't have those backup systems.
Also, here is a fascinating survival fact from the source material. People who are heterozygous for PK deficiency actually have an increased resistance to the most severe forms of malaria. It's a wild example of how a genetic mutation that causes disease can sometimes offer a unique, unexpected evolutionary edge.
And that brings us to Section 5, Memory Aids and Review. Let's lock in the highest yield concepts with a quick active recall check. First, which molecule acts as the feedforward activator of pyruvate kinase? And second, which hormone causes its covalent inactivation in the liver? Got it? The internal factory signal calling ahead is fructose 1,6-bisphosphate, and the hormone acting as our external padlock during a fast is glucagon.
I want to leave you with a conceptual question to ponder. Why does the liver inhibit pyruvate kinase during fasting while other tissues do not? It really highlights the incredible self-sacrificing design of human physiology, shutting down its own glucose burning to push carbon backward into gluconeogenesis, keeping the rest of your body, especially your brain, alive. That wraps up our explainer on pyruvate kinase, but it definitely makes you wonder, what other hidden metabolic pathways are secretly sacrificing themselves to keep you running right now? Keep that curiosity going, and thanks for joining me.
Hi everyone. So we've successfully followed glucose through the 10 reactions of glycolysis to finally produce pyruvate. But here's the thing, pyruvate has reached a fork in the road. Its journey isn't exactly the same for every cell or every situation. Today we're looking at what happens when the oxygen lights go out. We'll explore how your muscles keep working during a sprint, why heart muscle is just so good at eating lactate, and how yeast turns sugar into alcohol. Let's jump right in and look at the anaerobic fates of pyruvate. Now, you might be wondering, what actually happens to pyruvate when those oxygen lights go out? Well, it basically depends on 3 key things. First, the organism. Are we talking about a human or a microorganism like yeast? Second, the tissue. Is this happening in a sprinting muscle or a beating heart? And third, the redox state of that tissue, which is really just a fancy way of asking, is there enough oxygen around? Here's our roadmap for today's explainer. We'll cover the fork in the road, the NAD+ emergency lifeline, lactate in different tissues, lactic acidosis and shock, microbes and alcoholic fermentation, and we'll wrap up with high-yield memory aids.
All right, Section 1, the fork in the road. In a perfect world, right, with plenty of oxygen, what we call aerobic conditions, pyruvate just happily enters the mitochondria, it gets completely oxidized for this massive energy payoff. But the world isn't always perfect. In systems like mature red blood cells, which literally have no mitochondria at all, or in your skeletal muscle during intense, breathless exercise, the cell is forced into anaerobic glycolysis. Pyruvate simply undergoes a reduction reaction to form lactate, really just to survive.
Moving on to Section 2, the NAD+ emergency lifeline. Let's look closely at the actual molecular mechanics of this rescue operation. This conversion is catalyzed by an enzyme called lactate dehydrogenase, or LDH. Picture your 3-carbon pyruvate molecule. It has a ketone group that's a carbon double bonded to oxygen right there on the middle carbon. The LDH enzyme comes along, takes 2 hydrogens from a high-energy molecule of NADH and sticks them directly onto that middle carbon. This turns the ketone into an alcohol group, and boom, the result is L-lactate. Now, you've got to ask yourself, why does the cell even do this? Honestly, it seems like a terrible waste to spend that high-energy NADH we worked so hard to make earlier. But remember the NAD+ bottleneck. Reaction 6 of glycolysis, handled by G3P dehydrogenase, absolutely must have NAD+ to function. In anaerobic conditions, the electron transport chain isn't there to recycle NADH. Therefore, the reduction of pyruvate to lactate acts as the cell's emergency recycling center. It turns NADH back into NAD+ so that glycolysis can keep running and producing at least some ATP to keep the cell alive. It's a total lifesaver. Okay, section 3, lactate in different tissues. Let's zoom out to the macro level and see how different organs handle this in an elegant system of supply and demand. In your muscles during a full-on sprint, NADH production goes into overdrive, way more than the mitochondria can handle. This creates a really high NADH to NAD+ ratio. Lactate builds up, dropping the intracellular pH, which can lead to those incredibly painful muscle cramps we've all felt. However, lactate isn't just waste. It diffuses right into the blood and travels to other tissues. The direction of the LDH reaction actually depends on that NADH to NAD+ ratio. In organs like the liver and heart, this ratio is very low, so they actually reverse the reaction and oxidize lactate back into pyruvate. The heart is absolutely amazing in this regard. It almost exclusively eats that recycled pyruvate for energy via the TCA cycle. It's the ultimate lactate sink. The liver, on the other hand, takes that pyruvate and either burns it or uses it to build brand new glucose through a process called gluconeogenesis, just to send it right back out to those working muscles. It's a brilliant recycling loop.
Section 4, lactic acidosis and shock.
So what happens when this localized rescue operation scales up to a catastrophic systemic failure? Well, if your circulatory system collapses, your tissues suddenly stop getting oxygen altogether. Without oxygen, ATP synthesis in the mitochondria completely fails. To survive those critical minutes, your cells switch entirely to anaerobic glycolysis. This results in a massive flood of lactic acid pouring into the blood, creating a severe, life-threatening condition called lactic acidosis. We're talking about really grave clinical triggers here. Things like a myocardial infarction, a heart attack, a pulmonary embolism, uncontrolled hemorrhage, or just general shock. These completely prevent tissues from getting oxygen, forcing an extreme reliance on anaerobic ATP generation. While it's incredibly dangerous, producing even meager amounts of ATP through this pathway might just be life-saving during the time it takes to reestablish adequate blood flow. Because of this, doctors in the ER are constantly measuring blood lactate levels in shock patients. What they're looking for is something called an oxygen debt. This is the extra oxygen the body needs to recover and essentially repay the energy it utilized while oxygen was low. Closely monitoring these lactate levels is an absolutely vital tool for tracking a patient's recovery. Section 5, microbes and alcoholic fermentation. All right, let's totally switch gears from human medicine for a second. How do microorganisms, like yeast, handle this exact same suffocation problem? Well, they don't make lactate. Instead, they perform alcoholic fermentation in 2 distinct, irreversible steps. Step 1 is decarboxylation, an enzyme called pyruvate decarboxylase, which, by the way, vitally requires thiamine or vitamin B1, violently snaps a carbon off pyruvate, releasing a molecule of CO2 and creating acetaldehyde. Step 2 is reduction. Alcohol dehydrogenase then reduces that acetaldehyde to ethanol, using up an NADH in the process. You can see the 3-carbon pyruvate molecule literally lose its top carbon as a CO2 bubble. The remaining 2-carbon acetaldehyde is then hit with hydrogens from NADH to form ethanol. Just like the human LDH reaction, the whole point of making alcohol isn't to get the yeast drunk. It is strictly to regenerate NAD+, so the yeast can keep performing glycolysis. Finally, section 6. High-yield memory aids. To wrap things up, let's lock this into your memory for exam day. Think of NAD+ as the oxygen mask of glycolysis. If the cell is suffocating in an anaerobic environment, it uses LDH as a CPR maneuver to pump that NAD+ air back into the system so the cell doesn't die. For alcoholic fermentation, just remember bubble and brew. Bubble for the CO2 and brew for the ethanol. And never forget you need vitamin B1, thiamine, to get the bubble started. Before we go, I want to leave you with a conceptual question to ponder. Why is the regeneration of NAD+ considered the primary purpose of the LDH reaction in anaerobic conditions? And why exactly is heart muscle considered a sink for the lactate produced by skeletal muscle? Think about how the NADH to NAD+ ratio truly dictates that incredibly fine line between cellular energy and total collapse. What is oxygen debt, and why is it such a significant marker in shock patients? Dig into these concepts and you'll really master the material. Thanks for joining me on this explainer, and keep questioning the science around you. Hi everyone, and welcome back to the explainer. So we've spent the last few lessons trekking through the 10 granular enzymatic steps of glycolysis, looking at the fates of pyruvate, and even exploring some clinical defects along the way. But today we're zooming out. We really need to look at the big picture here. we're going to dive into the final bank statement of energy for the cell and see exactly how glycolysis serves as this massive central hub for building the rest of the body's molecules. Consider this our grand wrap-up of the Embden-Meyerhof-Parnasse pathway. Let's get into the final accounting.
Okay, let's dive into our roadmap for today. We're covering 1. The final bank statement. 2. Aerobic versus anaerobic glycolysis. 3. The biosynthetic supply depot. 4. The global glycolysis map. And we'll wrap up with 5. Our final exam review.
Section 1. The final bank statements. First up, let's look at the energy yield. Now, while we definitely made some ATP during this pathway, it's absolutely crucial to remember that the end products, whether that's pyruvate or lactate, they still contain the vast majority of the energy that was originally packed into that single glucose molecule. We're actually going to need the TCA cycle to fully squeeze that remaining energy out later on. But let's break down the ATP accounting right here in glycolysis. Think of it like a financial ledger. For our gross ATP gain, we produce 2 ATPs in reaction 7 and another 2 ATPs in reaction 10. So that's a total gross of 4 ATPs. Awesome, right? Well, remember our ATP utilization, the cost of doing business. We had to spend 1 ATP in reaction 1 with hexokinase and another 1 ATP in reaction 3 with PFK1. That's a total loss of 2 ATPs upfront in the investment phase. So when you do the math for our net gain, 4 minus 2 equals a rather meager but vitally important 2 molecules of ATP per glucose molecule.
Moving right along to section 2, aerobic versus anaerobic glycolysis.
So the fundamental difference between aerobic and anaerobic glycolysis? It essentially comes down to how we handle NADH. In anaerobic conditions, say in an intensely exercising muscle or inside your red blood cells, there is literally no net production or consumption of NADH. We make it in reaction 6 and then immediately use it right back up in the LDH reaction to reduce pyruvate and make lactate. Because of that, your net profit is just those 2 ATPs we just calculated. However, in aerobic conditions, things get a bit more exciting. We still net our 2 ATPs, sure, but we also produce 2 molecules of NADH. And you can think of these NADH molecules as high-value coupons that get sent directly to the electron transport chain, or ETC, inside the mitochondria. And the payout? Each NADH can be cashed in for approximately 3 ATPs. But here's a bit of a conceptual puzzle for you to chew on. If NADH is produced way out in the cytosol during glycolysis, how does it actually reach the electron transport chain, all the way inside the mitochondria?
Well, the answer comes down to some very strict cellular border control. The catch is that NADH cannot physically cross the inner mitochondrial membrane. It is completely impermeable. No way it's getting through. So because of this, the cell has to use specialized substrate shuttles to essentially teleport those high-energy electrons inside to the ETC. Meanwhile, it leaves the empty NAD+ behind in the cytosol so that glycolysis can just keep on running. It's a really elegant solution.
Which brings us to section 3, the biosynthetic supply depot. Okay, so the crucial point I want you to take away here is that it's a super common mistake to look at glycolysis and think it's only about making ATP. Yes, it degrades glucose to generate energy for sure, but it also acts as a massive biosynthetic supply depot, literally providing the raw materials for the rest of the cell to function. Let's look at the fates of 3 key intermediates. First up, DHAP from the cleavage phase. This provides the exact glycerol moiety we need to synthesize triacylglycerols, meaning our fats. Second, we have pyruvate from our final step. Pyruvate isn't just fuel. It can be turned into the amino acid alanine, it can be used as a substrate for gluconeogenesis to build brand new sugar, or it can be used to replenish the spinning wheels of the TCA cycle. Finally, there's glucose 6-phosphate, which sits at a highly critical junction. It can continue down glycolysis, sure, but it can also be diverted into the pentose phosphate pathway or even packaged away for glycogen synthesis.
To really cement this in your mind, I want you to think of glycolysis as both a local power plant and a supply depot rolled into one. It provides a little bit of immediate electricity, those 2 ATPs. And in an emergency, like a purely anaerobic state, it keeps the lights on just enough for the cell to survive. But its most important job in a healthy, well-oxygenated cellular city is providing the raw materials, like DHAP for fats and pyruvate for proteins, so the rest of the city can actually grow and build.
Section 4, the global glycolysis map. And this brilliantly illustrates exactly what we've been talking about. This final summary chart pulls together everything we've covered into one massive flow chart. On the left side, the metabolic side, we see it occurs in all tissues, right in the cytosol, turning out NADH and ATP. From pyruvate, it branches into the aerobic path, which remember requires oxygen and mitochondria to hit the TCA cycle, and the anaerobic path, happening in red blood cells or in exercising muscle to produce lactate. Then, shifting over to the right side for regulation, insulin in the well-fed state essentially give a massive green light to our three key regulatory enzymes, hexokinase, PFK1, and pyruvate kinase. Conversely, glucagon in the fasting state slap a red light on them via CMP and protein kinase A. Honestly, this one image perfectly captures the whole shebang of what we've studied so far. And finally, Section 5, Final Exam Review. Let's see what you remember.
Think about this conceptual question for a second. Why is the net energy yield of glycolysis considered meager compared to the total energy potential of a single glucose molecule?
Well, the answer takes us right back to our bank statement analogy. The end products, whether that's pyruvate or lactate, still contain the vast majority of the original energy locked up in their chemical bonds. Glycolysis really only scratches the surface. The TCA cycle is absolutely necessary to completely squeeze all that remaining energy out.
Next up, a quick recall question. Which specific glycolytic intermediate provides the glycerol moiety for triacylglycerol or fat synthesis? If you remembered our supply depot analogy, you know the answer is DHAP, or dihydroxyacetone phosphate, which is produced right in the middle of the pathway during that cleavage phase. Okay, let's step it up with an application question. During starvation, which metabolic hormone is dominant? And what is its exact physiological mechanism on hepatic pyruvate kinase? What is really fascinating here is how this brilliantly bridges biochemistry and physiology. During starvation, glucagon is the dominant hormone. It elevates intracellular CAMP, which then activates protein kinase A. Protein kinase A then goes on to phosphorylate and thereby completely inactivate liver pyruvate kinase. By shutting off this final step of glycolysis in the liver, it essentially prevents the breakdown of glucose and forces the liver towards gluconeogenesis, ensuring the brain gets the sugar it so desperately needs. You all did absolutely fantastic today. We've mastered the energy accounting, the supply depot dynamics, and the global regulation of glycolysis. But I want to leave you with this final provocative thought. If glycolysis, for all its intricate complexity, only yields a mere 2 ATP, what unimaginable power awaits when those final pyruvate molecules finally cross the border into the mitochondria? We are going to answer exactly that in our next explainer on the TCA cycle. Thanks so much for tuning in, and keep learning.
This has been Deep Dive Academy. Learn deep, remember forever.